This is a video about yeast. Let's start at the beginning. What are yeasts? Yeasts are a polyphyletic group of single-celled organisms characterized by having a nucleus and a cell wall. In this way, you can think of them as an odd blend between plants and animals. But they're not. If you thought that, you were wrong. Yeast are a totally distinct group of eukaryotes. And it is important to understand their inherent diversity, meaning their ecological functions and characteristics are equally diverse. Speaking of ecosystems, yeast are in almost all of them. In water, on animals like you, on dead stuff, and even on flowers. And in all of these ecosystems, yeast play an important role. They convert rocks into minerals, they combat pathogens and promote plant growth. But things start getting really interesting when you start looking at flowers. Why? Because of bees. And who doesn't love bees? I mean, just look at them. But in all seriousness, there is quite a lot of evidence to point towards the fact that bumblebees benefit directly and indirectly from consuming nectar that has been colonized by yeast. And after all this, it should be abundantly clear that yeast are very important in all imaginable ecosystems. However, the fact of the matter is, especially in this region of the world, there hasn't been that much research on these topics specifically. And the first step would be to identify these individual yeast species and find out what their characteristics are. But how would I, a mere student, go about identifying an entire subcategory of a kingdom of species? And I can feel you all behind the screen doubting me, thinking, Pitt, there is literally no way. And you would be completely right. This is a mammoth task that I, by myself, cannot complete. So let's not get our expectations too high. So what is the Wild Yeast Project? Well, it consists of 3 steps. Firstly, we must find the yeast. Secondly, we must identify the yeast. And thirdly, we must characterize them. The finding of the yeast is pretty simple. We go out into the environment and swab some fruits until we get some yeast. The identifying part is much harder. So how do we do that? Well, you can't just look at them. Even if you can see them under a microscope, turns out they're very small and all look pretty much exactly the same, regardless of species. A matter of fact, there is only so many ways you can arrange a blob of organelles. So, how do we do it? Well, since we can't look, we need to find other ways. And there is a solution. This solution lies in DNA. Now get ready for the boring bit, where I talk about sciencey sounding stuff for a couple of minutes. Here we have our yeast DNA. This small piece is representative for the ITS region. This region is present in all yeast, but is slightly different for each species. As you may know, a piece of DNA is linked in the middle, like this, by hydrogen bonds. These can easily be broken by temperature, known as denaturation. During denaturation, the 2 halves are split in 2, giving us 2 complementary pieces of DNA. We then move on to the annealing step. Here, primers are added. These tell an enzyme known as DNA polymerase to start copying the old strand. This means they will attach to the primer and start reading these base pairs, one by one, and adding the complementary base pair to that one. Meaning, if this were an A, the DNA polymerase would add a T, and so on, until we have copied the entire sequence. This step is known as the elongation step, where the DNA polymerase will do just that until the entire sequence of DNA has been filled. It is now important to realize that the sequence we have here, with our 2 sides, is identical to the initial piece, aside from the primer. Same goes for this piece. These are identical, and they are identical to the initial piece, meaning we have now, from 1 piece of DNA, created 2 pieces of DNA. This can be repeated, giving us 4 pieces repeat it again to give us 8, then 16, then 32, and so on. In other words, this is exponential growth, meaning we can have almost infinite amounts of DNA. These 3 steps, the denaturation, the annealing, and the extension, are known as a PCR, which stands for polymerase chain reaction. Using this method, we can get many copies of our initial DNA, which means we now have enough DNA to do the following steps.
As my colleague mentioned, the ITS region is different for each species. However, the lengths of the total region are quite similar, so we need to cut the region into pieces using enzymes.
Thank you very much. Yes, that is completely correct. The ITS region is similar for each yeast species, meaning it is not enough to simply replicate the DNA. We cut it into pieces using enzymes, meaning each species now has a different selection of cut pieces of DNA. When we look at this in something known as a gel, we can see these individual lengths of DNA as bands. This means by cutting the DNA into small pieces, we can give each DNA profile, so each yeast species, an individual barcode. An example of that would look somewhat like this, where we have 3 different yeast species which are identifiable by their unique barcodes. Sadly, the PCR process presented many problems. Often the PCR simply wouldn't work and we would fail to get good products. This meant much of our year was spent troubleshooting instead of really finding any solutions. So, we only got to the enzyme digest 2 times and neither of those were successful. In conclusion, the wild yeast project isn't quite done yet. I hope in the coming years we will continue our endeavour and hopefully someday finish it. However, in the meantime, there is still a lot of work to be done, and I hope that work will be continued once I'm gone. Think about it, it could be you. The glory is up for taking. But genuinely, this is important work. And even though we may have fallen short of our admittedly quite ambitious goals this year, the Wild Yeast project so far has been by no means a failure. The fact that you guys were forced to listen to me talk about yeast for more time than you ever will for the remainder of your life is a win in of itself. So, take care, don't forget about yeast, and bye-bye. Bye-bye.